All right. Welcome, everybody. Thank you so much for being here and being part of our seminar series. My name is Cinnamon Moffat, and I will be your host for this event. For anybody, I'm looking around, most of you have been here before. This is a hybrid event, so that means we have folks online and folks in the room. For the several folks that are online already, if you have any questions or technical issues, please use the chat function, and our volunteer, Roseanne, will read out those questions or address any issues you might be having. So feel free to communicate with us using that chat function. For folks in the room, when we get to question and answer, as you all know well, just raise your hand and I'll bring you this mic, and that way folks online can hear you and interact with us in the seminar setting. I've got a couple of quick announcements to give us before we get rolling for today. If you happen to be here at Hatfield, we do have a tsunami walk next week on March 19th. We're heading up to the Oregon Coast Community College Assembly site at 11. Once we're there, we'll have somebody meet us and talk a little bit about the new high ground housing if you are interested in that. Anybody is welcome to participate, but we'll be leaving Hatfield at 11 on March 19th. Our next seminar is on March 20th, where we have Diego Melgar, who is from the Department of Earth Sciences at the University of Oregon. He was scheduled before and had to reschedule with us, and he's going to be talking about how do we know the details of past Cascadia earthquakes. So I'm hoping that you can join. I want to pause here and just let folks know that if we go through a government shutdown, there might be some changes to our seminar schedule. I'll do my very best to let you know if anything changes or gets canceled. Hopefully on the Monday before the talk, but just kind of keep track. There might be some shifts. Hatfield's an amazing braided environment. We have lots of federal and state speakers. And so the good and the bad of that is that it might impact our seminar series, but I'll do my best to keep you informed. But I wanted to let you know that there might be some shifts. But Diego should be set for March 20th, so we should be good. Also wanted to remind folks that if you are a researcher here and you would like to table at Marine Science Day, the deadline for requesting a table is also March 20th. And if you are not a researcher here, but you are interested in coming and learning all about the different sciences, come on April 12th. That is when Hatfield opens its doors and we celebrate everything about science, and we invite everybody in to come see our open house. So Marine Science Day is coming. Yeah, if you have any questions about our events that are coming up, go ahead and go to the HMSC website, go to the events page, and you'll find everything you need to know to join us either in person or online at any of these events. But I think that's it for my announcements. So I want to jump into today's talk. Let me introduce today's speaker. We have Jingwei, so I'm going to mess it up again, Wei, who is a native from Shanghai, China. He obtained his bachelor's in biology and German from Wabash College in Indiana, and then he studied marine fisheries genetics in Virginia Institute of Marine Sciences and obtained his PhD in 2020. He did his postdoctoral work at Whitney Lab in Marine Biosciences at the University of Florida, and I believe he's going to talk to us a little bit today about that work. And since 2023, he has been a research associate here at Hatfield, working with the Oyster Genetic Project related to sex determination, sterility, and in collaboration with USDA and NOAA scientists. And so we're really excited to have him here. If you want to turn on your mic. Sounds good. All right, I'll hand the mic off to you. Thank you so much.
Thank you, Cinnamon. Hello, my name is Jingwei Song. I'm a research associate at Coombs here, and I collaborate with USDA scientists on oysters. But today we're not going to talk about oysters. This is the work from my postdoctoral work at UF. And you might be, curious what this animals you've been looking at. This is a single Hydractinia polyps. It's filmed sideways, so this is underwater. Yeah, back in 1973. It's a really cool video. So first, I want to give you some background where this work was conducted. It was at the Whitney Lab for Marine Biosciences. It is the marine lab for University of Florida. It is located just about an hour south of Jacksonville on the Atlantic coast of Florida. And then Whitney has a much smaller footprint compared to Hatfield. They have about 12 PIs total, and they have a long history of studying marine invertebrates such as ctenophores, nematostella, marine worms, and starfish. And then they're specializes in neurobiology and developmental biology, as well as stem cell biology. So Hydractinia is in the phylum of Cnidaria. It is a large group of animals consisting of jellyfish, hydroids, corals, and sea anemones. And then this group of animals are united by sharing a stinging cells called cnidocytes. So this picture on the left, this Pacific sea nettle, which you can see in the aquarium next door, are packed with these cnidocytes on the tentacles. So that's why it's so painful if you unfortunately get in contact with these tentacles. So what these cells are, they're essentially small pressurized cells. And you can see this electron microscopy image. It's a harpoon that can, upon touch, it can inject into the prey and release the venoms. So all the species in Cnidarius have that, but some are more toxic or venomous than others. But thankfully, Hydractinia's cnidocytes are not toxic. We can handle them without feeling any pain, really. And Hydractinia is a species that typically grow on the shell of hermit crabs. And you will see here people referring them to snail fur because their furry texture on the hermit crabs. And they form this symbiotic relationship with their hermit crabs. These Hydractinias, they're colonial. So the picture on the right, you can see I pointed out these feeding polyps. They're actually all belong to the same individual. And then they can share resources and they have coordinated movements. Yeah. So Hydractinia has been under studying for a long time. The first record of Hydractinia was by German scientist August Weismann back in the late 1800s. He was, and among other German scientists at the time, first coined the term stem cells in German. So the stem cells actually translation from the German. And his idea was, you know, there's this kind of cells, they give rise to all kinds of other cells, as the diagram shown in the middle. And then they have very meticulous hand drawings of these marine invertebrate species. So on the right, this looks like a Hydractinia cross-section. And then over the next 100 years, people continue to study them. And then we know a lot about its biology, and the life cycles are relatively short. So it can be completed 2 to 3 months in the lab, so which makes a really convenient organism to work with. They have separate sexes. So one colony is a male and one colony is female. So this diagram just shows you the life cycle from embryos to larvae and metamorphosis. The top right, you see a primary polyp. That's what you saw on the title slide. So this whole colony started off with one single polyps. And eventually this polyp will grow and expand to cover the entire hermit crab shells or whatever other surfaces you're growing on. I will show you on the next slide. And then different sexed colonies will have these stereotypical sexual polyps. So on the male colonies, you will only see this sexual polyps packed with sperm. And then the female polyps, you can see these eggs in these sac structure. So in the lab, we actually don't need to keep hermit crabs. How we keep them is these slide racks. This way we can keep relatively high densities of these animals. And if they grow really well, they can cover the entire slides in just a few months. And then we can do experiments with them. So this is a image of a young colony looking from above. Starting from the right, you have these young feeding polyps just starting to grow. You have a single mature feeding polyps here, contracted, extended, and then there's the structure, web-like structure we refer to stolonal mat. It's essentially a gastrovascular network that how different polyps are communicating and share nutrients as a whole colony. And then you have these stolon tips that are extending outwards as the colony expand. So this is a mature colony, and you can see the colony has grown and we have a lot more polyps. So starting from the middle, these are the oldest polyps. So the oldest part of this one organism, that's where you first start seeing sexual polyps. I don't know if you can quite see there. This is a female. And then towards the outer rim, you have many more feeding polyps, but fewer sexual polyps. So once we have a colony like this, we could induce them to spawn. So I will show you a video of females spawning. So the way we induce them to spawn is just to shine a very bright light on them. So it's relatively straightforward. So you can see these eggs are floating in this bin, right? That's how you tell you got pretty successful spawning. And then we can filter the water, just collect the eggs. If a healthy colony, we obtain hundreds of eggs every week or so. So it's really convenient. There's an egg just getting released. It's kind of hard to see here. All right, so I'll show you how a male colony is spawning. You will see it's very different because sperms are so much smaller. This is from a previous member of the lab, Schnitzer lab. You can see it's, I mean, if you work with shellfish and other marine inverts, it's kind of similar. You have these cloud of sperm in the water. You don't really see the granularity of the sperm like the eggs. But yeah. So here you can see a stream of sperm getting released from a sexual polyps. And we always get way more sperms than we actually need. So hopefully by now I've shown you, give you a good background of Hydractinia, why is an ideal research model, because for these various reasons. Also, I should mention it has a sequenced genome, which is not uncommon these days. Also, this Hydractinia is amenable to genetic manipulations, such as transgenics, CRISPR-Cas9. So this green image on the right shows you a transgenic Hydractinia that every single cell is expressing green fluorescent protein, so which makes very easy to track individual cells and cell types under development. So now I want to switch gear to talk about regeneration. So, excuse me. So we all know this is a movie from Spider-Man, right? There's a, if you haven't watched it, it's a mad scientist, was an amputee, and he wanted to regrow his arm, basically, but things went wrong and turned him into a lizard monster. So that's not what we're talking about today. So in our humans, I mean, eventually it's a cool idea, right? If we could help using and leveraging the knowledge from these animals that can actually regenerate and then apply it to humans. But in adult humans, we have limited stem cells that are low in numbers and then can only differentiate into certain types of cells, as shown on the right. However, some other vertebrates have amazing regenerative potential, such as the Mexican axolotl, which they could regrow their limb after it being cut off. So many inverts have extensive regenerative potentials, way more impressive than even the axolotl, but they're also anatomically relatively simple organisms. For example, this one, the freshwater flatworms, planarian, and then these sea stars we can find just out here in the bay on the jetties. These sea stars, if they are cut into pieces, every single piece can regrow into a new individual. And then you have Hydra. Hydra is a freshwater relative to Hydractinia, also had a very long history of studies. This is just one more example of planarians. You can see the top one planarian, if you cut it into 3 pieces, every single piece will regrow into a new individual, which is kind of freaky. Because in the middle, you can see like even regrows a new head with the 2 eyes. Hydractinia can also regrow a head, but if you cut the head off, the top part doesn't regrow really, but the bottom half can regrow a head in about 3 days. This is just a time-lapse video of the head regeneration in Hydractinia. You can see in the end, this animal is kind of moved out of focus a little bit, but hopefully you can still tell the head, the tentacles are getting regenerated. So how do they achieve this amazing regeneration event? So not all stem cells are created equal. So some stem cells are pluripotent, meaning they can give rise to all the cells. So I like this cartoon here. It says stem cell parental advice. You can be anything you want to be when you grow up. That really is referring to pluripotent stem cells. Then you have multipotent stem cells, which are have more limited potential to give rise to other cell types. For example, in humans, in the bone marrows, you have bone marrow stem cells, which can give rise to different kinds of blood cells. But that's all these bone marrows can give rise to. So that's what I mean by multipotent. And Hydractinia has these stem cells we call eye cells because they're interstitial. They're kind of lodged in between the tissues. I'll refer to these stem cells as eye cells interchangeably from now on. These characters give eye cells, make people easy to identify them. And then because they're ribosome rich, you can stain them with different dyes. So what we know is their shapes, their general location is on the lower half of these polyps. So what you see here are 4 genes that are human stem cell homologs that we found in Hydractinia, which they all more or less shows these stem cells are on the bottom of the Hydractinia. So here you have a Hydractinia during head regeneration that a fluorescently labeled eye cell that's migrating towards where the head was cut off. So you have these cells the stem cells are involved in head regeneration, and they also migrate to where they need to be, essentially. So collectively, we know eye cells are pluripotent. They can give rise to all the cell types in regeneration. But what about individual eye cells? So there are 2 competing models that one is the pluripotent one. So on the left, you can see these eye cells, not only they can self-renew, as in these circular arrow, so self-divide, but they can also give rise to different kinds of cells. Another model is not only these eye cells self-renew, they have a subsection of the eye cells, they're only multipotent, so they can only give rise to specific sets of progenitor cells, as you can see the group by the color here. So how do we tackle this question is to use single-cell RNA sequencing. So traditionally, bulk RNA sequencing, you have a tissue, whatever it might be, you grind it up as your input, and then you can sequence all the genes in the sample. However, all your heterogeneity is masked because you grind them up. Alternatively, maybe since the last 10 years or so, we have a technology called single-cell analyses. So you dissociate your tissue to obtain these individual cell types in cell suspension, and then you sequence these individual cells separately, and then you can reveal your heterogeneity in your tissue samples. So that's what we did. We took Hydractinia, a mature colony. We cut out 10 feeding polyps and then dissociate them using enzymes. So under a microscope, you can see this diversity of cell type, cell size, and we could tell which ones are which. So we're confident going in, our samples had a good mixture of different cells. I was skipping forward all the details on the sequencing, but essentially we did a UMAP projection of this data. We've received about 50,000 cells. Every dot is a cell on this map. And then the clusters are cells with similar gene expressions, which could correspond to cell types. Next, we went on to do more validation. Before that, I want to talk about, the cellular layout of Hydractinia, right? It has a gastrodermis, which is the lining, facing the gastroderm. And then you have another cell layer, epidermis, which is facing outside the outside environment. And then we have generally a good idea of where different cells are, thanks to the microscopy and all the cell biology from the last century. And then we recover these 5 major cell types in Hydractinia, as shown by the different colors on the right picture. So we have this green cnidocyte development series with eye cells, neurons, epithelial cells, gland cells. And we actually was quite surprised. We found an unknown cluster that we're still not quite sure what they are. So we used HCR, fluorescent in situ hybridization, for the marker genes for each cluster. We're trying to validate where these cells are in an actual animal. So what this tool does is you can query a single gene and design probes to the DNA sequence, and then it will hybridize to where these genes are expressed. And then you can multiplex different fluorescent, so you have different genes in the same animals. It's actually quite convenient to locate where different genes are expressed. So first we'll look at the cnidocyte development trajectory because it's the largest feature we have on the UMAP. From this right picture, we have eye cells, and then the greens are the cnidogenesis, which is the cnidocyte development timeline, if you will. And then we were able to identify genes along this trajectory So for example, highlighted here, TXD12 and nematocillin A, we know nematocillin is a gene only present in mature cnidocytes. As you can see here, we used the HCR, nematocillin A nicely showed on the tentacles, where we know that's where the cnidocytes are packed. We have the highest density of them. And then TXD12, they're showing up on the lower half where the eye cells are. Yeah, so this is just a good sanity check that our data set is actually meaningful. And then we can just extract the cnidocyte cells and recluster it. This is just a different look of the UMAP you've seen before. We still have two branches of the cnidocytes. So we're curious if these two branches correspond to different cnidocyte subtypes. And the evidence seems to suggest that. So for example, the magenta color shows these cells are generally common along the tentacles. However, this yellow color on the other branch seems to be restricted on the tip of the tentacles. So again, this tells us our UMAP is biologically meaningful. Next, I want to show you some neuron. From a previous study in the 1970s, we know there should be two types of neurons. One was called ganglion cells, one is called the sensory cells. And then we did find two clusters with some classic neural markers. And then the same thing, we use HCR to see where these cells are located. So for the ganglion cluster, we use a gene called RFMide, which is a classic neuropeptide gene. And as you can see, it's concentrated in the head area and more sparsely in the rest of the polyp. And another, for the sensory cluster, we chose a gene we call MIP-like. And you can see it's more concentrated on the tentacles and not so much, almost entirely absent from the head and some on the tentacles. So this picture is just a overlap of the two markers. So we have good evidence that these two neural markers, neural clusters, are labeling two different types of neural cells. So I'm not going to talk about all the clusters, but I want to talk about the eye cells. And then so we're curious if eye cells has these subpopulations. So we took the eye cells out, did a subclustering analysis. What we found was there's different subpopulations. So we have the classic eye cell inside these eye cell cluster characterized by these genes. And then we have neural progenitors and then cnidocyte progenitors. But we also have cells that we couldn't really annotate which progenitors there actually are. So this is still a work in progress. So far, this is our new working model for eye cells and cell development. So we have these pluripotent eye cells. They can self-renew, and then they can give rise to lineage-primed progenitor cells. And then these progenitor cells can give rise to differentiated cells. So in summary, Hydractinia is an exciting cnidarian research organism. And we've used single-cell RNA-seq to study all the Hydractinia cell types. And then we're also gaining new insights into cell composition and dynamics. So this is a huge data set, and we hope this can be very helpful for the community to use. It's just useful for other people as well. With that, I would like to thank these collaborators from UF and NIH. Happy to take questions.
Thank you so much. So we're going to switch to questions, and we'll go from questions online to questions in the room. Roseanne, any questions online yet? Okay, for folks online, if you have any questions, go ahead and put them into the chat. How about in the room, everybody? Any questions? There we go.
Very, very nice talk. What do you feed these critters on?
Brine shrimp.
Ah, and
Yeah.
have you done any work to look at the development of digestive cells?
The gland cells.
Mhm.
Yeah, so far we did HCR on those cells. We can tell they're in the gastrodermis. Their, morphology and internal structure are kind of what we expect, but we haven't done much further, test with them. We can just tell we know where they are. It seems to confirm what we know. Yeah, but there's more experiment you could do with them for sure.
Thank you.
Great talk. So I know these critters are quite small, but is there any thought of doing spatial RNA versus single cell RNA so you can get an idea of actually where in physical space the various gene expression is going on?
So the question is, have we thought about doing spatial transcriptomics? Because this is single cell,
Yeah.
but it's like, yeah, I see what you're saying. Yes, we thought about it, but no, it's too expensive. But I can tell you at Oysters, what we're working right now, we're actually looking at spatial transcriptomes. And hopefully next year I'll present some of that. Yeah,
Well, I look forward to it.
thanks.
Other questions in the room? Okay, give me a sec. Need to wiggle in front of a couple of you. Thanks.
Nice talk. I enjoyed it very much. I remember in undergraduate invertebrate zoology, we used to have cells that of what was it the now it's skipped out of my mind. Sponges. You'd mix sponges. You'd separate the sponges, the cells from the sponges. Then they would aggregate together. If you gave them time. Do these cells move to aggregate after you separate them?
Right. In Hydra, which I showed a picture of, the freshwater cousin of Hydractinia, they've shown they would aggregate, like the Terminators, you can scramble them up, and then over time, some will reaggregate and reform the body plan and actually really just come back as an individual. Hydractinia, I'm not aware anyone tried that. Maybe we should try it, blend them up.
All right, looks like we got a question online. Go ahead, Roseanne.
You mentioned that they grow on hermit crab shells. Is it certain species they grow on or all species?
That's a very good question. The species we work with is called Hydractinia symbiolongicarpus. So that is growing on a specific type of hermit crabs. But in Hydractinia, there are over 30 species worldwide. So I'm sure there's, other Hydractinia species only grow on other hermit crab shells.
All right, other questions? Okay, hang on, everybody. Got to get to the back of the room, and then I'll get to the front of the room.
Great talk as well. I was just wondering in terms of your single cell sequencing, when you were doing your sorting and you're sorting through your cell types, about how many were you getting that you were bringing into the 10X per cell type, and were you using multiple organisms or able to collect from, they're pretty small. So I was like, I don't know how many you're getting. And if that was per individual or multiple individuals per cell.
Right. We use the same colony, but 10 individual feeding polyps. Just use the enzyme to break them up, digest it, but we didn't use all of it. So we have an excess of cells that we have, but then we took a subsample. I actually counted the cells. And then we're trying to target a specific certain number of cells to go in the experiment. So here we have around 50,000 cells on the UMAP, but I can't remember on top of my head, but we definitely put more in to begin with. Does that answer your question?
Yeah.
All right, now I'm gonna go to the front of the room, so you get to hear me tromping around and up and down the stairs again, everybody.
Very nice talk. Very interesting. My first question is an ecological one. You mentioned that there are separate sexes, but are the colonies on an individual hermit crab have a colony of males and a colony of females, or do they need to, have the hermit crabs close enough for reproduction to be successful? And then I do have a second question. You talked about the similarity to, some of the stem cells to human stem cells. And where do you think that work is going in the future? Just curious. Okay.
Yeah, the first question, if there's multiple colonies with different sex on the same hermit crab, it's definitely possible, because depending on where the larvae land, there could be, two larvae with opposite sex land on the hermit crab. But then there's actually another interesting fact about Hydractinia I didn't touch on. It's called allorecognition. So what happens is two different colonies, if they're genetically related, they can fuse under certain circumstances and become one. But if they're more distantly related, they will actually fight each other when the stolen tip touch, and then they will reject each other. So, and eventually there will be one colony actually dominates the other. So in that case, I guess it's possible on the same hermit crab shell, you have two different individuals, but they can coexist if they're closely related. Yeah, but yeah, we need to look closer in that. I would actually do experiment with it. And the second question is, how's the stem cell, the work in Hydractinia can translate to human?
With the similarity that you mentioned to human stem cells, I think you said there were 4 different ones that were somewhat similar to human stem cells, if I understood correctly.
Those images I showed, there are only 4 homologs, but actually people have looked at, there's way more genes in these stem cells or eye cells in Hydractinia share homologs with human stem cells. So I guess the idea is, if the toolkit, right, Hydractinia, human, and other species, they're not that different. It's just, how they are regulated are different. So why does, human cannot regrow a head, but Hydractinia can? So maybe by studying Hydractinia, we can hopefully eventually, learn something why human can. Yeah.
All right. Thank you.
Can I go back to the cells or the patches merging? If you had a male and a female patch on a hermit crab's back, you said that if they were genetically related, they could possibly merge. Is there a dominant sex then, male or female?
That I'm not so sure. Yeah, I think there's one lab in the world that's expert in that. Yeah, I can get you connected.
Thank you. Just curious. Any other questions in the room? Yeah, hang on.
Hello, thank you for the talk. It's wonderful. I was just wondering if any of the cells or the critters themselves, do they respond to any pain stimuli?
Pain?
Or heat, or changes in temperature, water temperature, or anything of that sort?
Yeah, I mean, they're living organisms, right? I would guess they're definitely, if they're too hot, too cold, not eat enough, different stressors in the marine environment. But so in our lab, we're trying to keep them as happy as they can, right? The room is temperature regulated. The water they're growing is always the same salinity. They're always very well fed. So the goal is to keep them, as healthy as they can so they can keep reproduce. Right. I think if we don't take care of them, they will start to wither away and die. But they're pretty hardy. They're hard to kill. So that's another, reason people study them, because they're not, too picky, but we still want to keep taking good care of them.
All right, last call for questions, everyone. Roseanne, any other questions online? All right, let's go ahead and wrap up there and let you off the hook. Thank you so much, everyone, for being here. I hope I see you next week, and let's give one more round of applause.